Facilitated diffusion is a critical transport mechanism that allows certain molecules to move across cell membranes. Cell membranes form a protective barrier around cells. These membranes contain specialized transport proteins that create pathways through the phospholipid bilayer. Facilitated diffusion always moves substances from areas of higher concentration to areas of lower concentration. Unlike active transport, facilitated diffusion doesn't require energy. The molecules naturally move down their concentration gradient. Facilitated diffusion is essential for transporting molecules that cannot pass through the phospholipid bilayer on their own, such as water-soluble molecules, ions, glucose, and amino acids. The cell membrane forms a critical barrier that separates the internal environment of the cell from the external surroundings. This membrane is composed of a phospholipid bilayer with two layers of phospholipids arranged in opposite directions. Each phospholipid has a hydrophilic head that interacts with water and hydrophobic tails that avoid water. The membrane acts as a selective barrier, allowing some molecules to pass through while blocking others. Small nonpolar molecules, like oxygen and carbon dioxide, can easily diffuse through the hydrophobic core of the membrane. Small polar molecules, like water, can pass through but at a slower rate due to the hydrophobic nature of the membrane interior. Large molecules, however, cannot pass through the membrane due to their size. Charged molecules, like ions, are repelled by the hydrophobic environment and cannot easily cross without assistance. Despite the membrane barrier, cells need to transport essential molecules like glucose, amino acids, and ions. This selective permeability creates the need for facilitated diffusion mechanisms, specialized transport proteins that help essential molecules cross the membrane barrier. Concentration gradients are fundamental to understanding facilitated diffusion. A concentration gradient exists when the concentration of molecules differs between two adjacent areas. Let's visualize a cell membrane with a high concentration of molecules on one side and a low concentration on the other. The membrane contains transport proteins that allow specific molecules to pass through in a process called facilitated diffusion. According to the concentration gradient, molecules naturally move from areas of high concentration to areas of low concentration. This movement follows the second law of thermodynamics, which states that systems tend toward maximum entropy or disorder. A key feature of facilitated diffusion is that it requires no energy input. The movement is driven by the concentration gradient alone, making it a passive transport process. Transport proteins are the key components that enable facilitated diffusion across cell membranes. The cell membrane consists of a phospholipid bilayer that forms a barrier to most molecules. Transport proteins have several important features that allow them to facilitate the movement of specific molecules. These specialized proteins are embedded directly within the cell membrane. Each transport protein has specific binding sites that recognize and bind to particular molecules. After binding, transport proteins undergo a conformational change, altering their shape to move substances across the membrane. This elegant mechanism allows molecules to pass through the membrane without disrupting its structural integrity, which is crucial for cell survival. Transport proteins are essential components that enable cells to precisely control which molecules can enter and exit, making facilitated diffusion possible. Channel proteins are specialized transport proteins that create water-filled passageways through the cell membrane. Unlike carrier proteins, channel proteins form permanent pores or tunnels that allow molecules to flow through the membrane. Channel proteins are highly selective, allowing only specific types of molecules to pass through. This selectivity is based primarily on the size of the molecules. Small molecules can pass through, while larger ones cannot. Additionally, many channels are selective based on the electrical charge of molecules. Let's look at two important examples of channel proteins found in cell membranes. Aquaporins are specialized channel proteins that allow water molecules to pass through the membrane efficiently. These channels are so efficient that billions of water molecules can pass through a single aquaporin each second. Ion channels are another important type of channel protein that allows specific ions to pass through the membrane. These channels are highly selective for specific ions such as potassium, sodium, calcium, or chloride. For example, a potassium channel allows potassium ions to pass while blocking other ions. It's important to note that many channel proteins are not always open. They can be regulated by gates that respond to different stimuli. Carrier proteins facilitate the movement of specific molecules across the cell membrane through a unique mechanism. Unlike channel proteins, carrier proteins undergo significant conformational changes during transport. Let's examine how glucose transporters, or GLUT proteins, move glucose across the membrane. First, the carrier protein has a specific binding site that recognizes glucose molecules. After glucose binds, the carrier protein undergoes a conformational change, rotating to expose the binding site to the opposite side of the membrane. The glucose molecule is then released into the intracellular environment due to the lower affinity of the binding site in this new conformation. Finally, the empty carrier protein returns to its original conformation, ready to transport another glucose molecule. GLUT proteins are a family of glucose transporters with several key features. Let's compare simple diffusion and facilitated diffusion, two fundamental processes for moving molecules across cell membranes. Simple diffusion occurs when molecules move directly through the phospholipid bilayer without any assistance from proteins. In contrast, facilitated diffusion requires specialized transport proteins embedded in the membrane to help molecules cross. Simple diffusion is limited to small, nonpolar molecules like oxygen and carbon dioxide that can slip between the lipid tails of the membrane. Facilitated diffusion, however, can transport larger or polar molecules like glucose and amino acids that cannot pass directly through the hydrophobic core of the membrane. Simple diffusion doesn't require proteins and is limited to small nonpolar molecules. Its rate is directly proportional to the concentration gradient. Facilitated diffusion requires transport proteins and accommodates larger or polar molecules. It works faster than simple diffusion, but can become saturated when all transport proteins are in use. When we compare their transport rates, simple diffusion shows a linear relationship with concentration, while facilitated diffusion demonstrates a saturation curve as transport proteins become fully occupied. To summarize, both types of diffusion move molecules from high to low concentration without energy input, but they differ in speed, specificity, and the types of molecules they transport. Let's compare facilitated diffusion and active transport, two fundamental mechanisms for moving molecules across cell membranes. Facilitated diffusion is a passive transport process that moves molecules from areas of higher concentration to lower concentration through specialized transport proteins. In facilitated diffusion, molecules move down their concentration gradient without requiring energy. The key characteristics of facilitated diffusion are: it's a passive process requiring no energy, it follows the concentration gradient, and it uses transport proteins to help molecules cross the membrane. In contrast, active transport is a process that moves molecules against their concentration gradient using energy, typically in the form of ATP. Active transport pumps use energy from ATP to move molecules against their concentration gradient, from areas of lower concentration to areas of higher concentration. The key characteristics of active transport are: it's an active process requiring energy from ATP, it works against the concentration gradient, and it uses specialized transport pumps. Let's compare these two transport mechanisms side by side to better understand their key differences. While facilitated diffusion requires no energy, active transport depends on ATP as an energy source. Facilitated diffusion always moves molecules down their concentration gradient, whereas active transport can move molecules against their gradient. The rate of facilitated diffusion is limited by the strength of the concentration gradient, while active transport can achieve higher transport rates. Both processes show saturation kinetics, meaning they can reach a maximum rate when all transport proteins are occupied. Facilitated diffusion is commonly used for glucose and amino acid transport, while active transport includes examples like the sodium-potassium pump and calcium pumps. The fundamental difference between these two transport mechanisms lies in their energy requirements and their relationship to concentration gradients. Facilitated diffusion relies on transport proteins to move molecules across cell membranes. Saturation is a key property that differentiates facilitated diffusion from simple diffusion. It occurs when all available transport proteins are actively engaged in moving substrates. At low substrate concentration, only a few transport proteins are engaged in moving molecules. As substrate concentration increases, more transport proteins become occupied. At high substrate concentration, all transport proteins are engaged, leading to maximum transport rate. Let's visualize this with a graph showing transport rate versus substrate concentration. In facilitated diffusion, the transport rate initially increases with substrate concentration. But as substrate concentration continues to increase, we reach a point of saturation where all transport proteins are occupied. This creates a maximum transport rate that cannot be exceeded, even if we further increase substrate concentration. In contrast, simple diffusion doesn't rely on transport proteins and shows a linear relationship without saturation. To summarize, saturation is a defining characteristic of facilitated diffusion. It occurs when all transport proteins are engaged, creating a maximum transport rate that cannot be exceeded. This distinguishes it from simple diffusion, which shows a linear relationship between concentration and transport rate without saturation. Glucose transport across cell membranes is a classic example of facilitated diffusion in action. Glucose is a vital energy source for cells, but it faces a challenge. It's a relatively large, polar molecule that cannot cross the hydrophobic cell membrane on its own. Without assistance, glucose would be unable to enter cells efficiently despite the concentration gradient driving it inward. This is where GLUT proteins come in. GLUT stands for glucose transporter, a family of specialized transmembrane proteins that create channels specifically for glucose to pass through. The transport mechanism involves a series of steps. First, glucose binds to a specific site on the GLUT protein. This causes the protein to change shape, moving the glucose through the membrane. Finally, glucose is released into the cell, and the protein returns to its original conformation. Different cell types have specialized GLUT proteins based on their glucose needs. GLUT1 is abundant in red blood cells and the brain, ensuring a constant supply of glucose. GLUT2 in the liver and pancreas helps with glucose sensing. GLUT4, found in muscle and fat cells, is unique because it responds to insulin, moving to the cell membrane only when insulin is present. The efficient transport of glucose is critical for cellular energy production. Once inside the cell, glucose enters the glycolysis pathway, eventually producing ATP, the energy currency of cells. This energy powers everything from muscle contraction to neuron firing to cell division. Understanding glucose transport has important clinical implications. In diabetes, GLUT4 proteins fail to respond properly to insulin, preventing glucose uptake in muscle and fat tissues. GLUT1 deficiency can cause seizures and developmental delays due to insufficient glucose in the brain. Cancer cells often increase GLUT expression to fuel their rapid growth. In summary, glucose transport via GLUT proteins is a perfect illustration of facilitated diffusion in action, demonstrating how cells have evolved elegant solutions to move essential molecules across membrane barriers. Ion channels are specialized membrane proteins that facilitate the diffusion of specific ions across the cell membrane. These channels are highly selective, allowing only specific ions such as sodium, potassium, or calcium to pass through. Many ion channels are gated, meaning they can open and close in response to specific stimuli. There are three main types of gated channels. Voltage-gated channels respond to electrical signals, ligand-gated channels respond to chemical messengers, and mechanically gated channels respond to physical forces. One of the most important examples of ion channels in action is the generation of action potentials in neurons. Neurons contain voltage-gated sodium and potassium channels that open and close at different times to generate electrical signals. During an action potential, sodium channels open first, allowing sodium ions to rush into the cell, causing depolarization. Then potassium channels open, allowing potassium ions to exit the cell, causing repolarization and a return to the resting state. To summarize, ion channels are specialized proteins that facilitate the diffusion of specific ions across cell membranes. They're critical for neuronal signaling, muscle contraction, and many other biological functions. Several key factors influence the rate of facilitated diffusion across cell membranes. Let's examine the four main factors that determine how quickly molecules move through transport proteins. The steepness of the concentration gradient is a primary factor affecting diffusion rate. A steeper concentration gradient, meaning a greater difference in molecule concentration between the two sides of the membrane, results in faster diffusion. The second factor is the number of transport proteins in the membrane. Membranes with more transport proteins allow for faster, facilitated diffusion. With more proteins, more molecules can pass through simultaneously, increasing the overall rate of diffusion. Temperature is the third major factor affecting facilitated diffusion rates. Higher temperatures increase the kinetic energy of molecules, causing them to move more rapidly. This increased molecular motion leads to more frequent collisions with transport proteins, accelerating the rate of facilitated diffusion. The fourth factor is molecular specificity, which refers to how well molecules match the specific transport proteins. Transport proteins are highly selective, only allowing molecules with the correct size and shape to pass through. Molecules that match the transport protein's binding site can pass through, while others cannot, regardless of the concentration gradient. To summarize, four main factors determine the rate of facilitated diffusion across cell membranes. First, the steepness of the concentration gradient drives the direction and speed of diffusion. Second, the number of transport proteins in the membrane determines how many molecules can pass through simultaneously. Third, temperature affects molecular kinetic energy and movement speed. And fourth, molecular specificity ensures only compatible molecules can utilize specific transport proteins. Understanding these factors helps explain how cells regulate the movement of essential molecules across their membranes. Temperature significantly affects how quickly molecules move through transport proteins in facilitated diffusion. At normal body temperature, around 37 degrees Celsius, facilitated diffusion occurs at an optimal rate. When temperature decreases, molecular movement slows down. Lower temperatures reduce the kinetic energy of molecules and decrease protein flexibility. As temperature increases, molecules move faster and transport proteins become more flexible. This increases the rate of facilitated diffusion. However, at extreme temperatures, above 50 degrees Celsius, proteins begin to denature. Protein denaturation alters the three-dimensional structure of transport proteins, causing the channel to collapse and preventing molecules from passing through. This graph shows how temperature affects the rate of facilitated diffusion. The rate peaks at normal body temperature around 37 degrees Celsius. At low temperatures, the transport rate is reduced due to decreased molecular movement and protein flexibility. At optimal temperature, facilitated diffusion reaches its maximum rate. As temperature increases above the optimum, the rate initially increases, but then begins to decline as proteins start to destabilize. At extreme temperatures, protein denaturation occurs, causing facilitated diffusion to essentially stop. To summarize, temperature affects facilitated diffusion in multiple ways. Higher temperatures increase molecular movement and make transport proteins more flexible, both of which enhance diffusion rates. However, extreme temperatures denature proteins and stop the transport process completely. pH plays a crucial role in facilitated diffusion by affecting the structure and function of transport proteins. Most transport proteins have an optimal pH range where they function most efficiently. This is typically around neutral pH for many cellular proteins. At optimal pH, the protein's structure allows molecules to pass through efficiently via facilitated diffusion. Transport efficiency follows a bell curve relationship with pH, with maximum efficiency at the protein's optimal pH range. When pH changes, it affects the ionization state of amino acids in the protein, altering hydrogen bonds and electrostatic interactions. In acidic environments, the increased concentration of hydrogen ions can protonate negatively charged amino acids, changing the protein's shape. pH changes directly affect binding sites by altering their shape and chemical properties. Acidic or basic conditions can disrupt the precise configuration needed for substrate recognition. In basic environments, hydroxide ions can deprotonate positively charged groups, again disrupting the protein's structure and function. Outside of the optimal pH range, transport efficiency decreases significantly. This is why maintaining proper pH in cellular compartments is essential for normal function. Understanding pH effects on transport proteins is critical in both normal physiology and in pathological conditions where pH balance is disrupted. Transport proteins are essential for proper cellular function, allowing specific molecules to cross the cell membrane through facilitated diffusion. When transport proteins malfunction, serious medical conditions can result. We'll examine 3 significant disorders: glucose-galactose malabsorption, cystic fibrosis, and certain forms of diabetes. Glucose-galactose malabsorption is caused by mutations in the SGLT1 transporter. In healthy intestines, SGLT1 facilitates absorption of glucose and galactose from food. With defective SGLT1 transporters, these sugars cannot be absorbed. The resulting buildup of sugars in the intestine causes severe diarrhea and dehydration, particularly in infants. Cystic fibrosis results from mutations in the CFTR gene, which normally creates chloride ion channels in cell membranes. These channels are critical for maintaining proper fluid balance. When CFTR channels malfunction, chloride ions cannot pass through properly. This leads to the production of thick, sticky mucus that clogs airways in the lungs and obstructs the pancreas and other organs. In diabetes mellitus, particularly type 2 diabetes, cells develop insulin resistance. Normally, insulin triggers the movement of GLUT4 glucose transporters to the cell membrane. With insulin resistance, this GLUT4 translocation is impaired, preventing efficient glucose uptake from the bloodstream. This leads to chronically elevated blood sugar levels and resulting metabolic complications. Understanding the molecular basis of transport protein disorders has led to targeted treatment approaches. For glucose-galactose malabsorption, dietary management is essential, eliminating glucose and galactose from the diet. For cystic fibrosis, breakthrough CFTR modulator drugs have been developed that can improve protein folding, trafficking, and function for specific mutations, addressing the root cause of the disease. In diabetes, medications like thiazolidinediones act as insulin sensitizers, improving GLUT4 translocation to the cell membrane and enhancing glucose uptake. Scientists use specialized laboratory techniques to study facilitated diffusion across cell membranes. The first major technique is radioactive tracers. In this method, molecules are labeled with radioactive isotopes, such as tritium or carbon-14. This allows scientists to track their movement across membranes and precisely measure transport rates. The second technique is fluorescent tagging. Transport proteins are labeled with fluorescent molecules like green fluorescent protein. Using fluorescence microscopy, scientists can visualize protein locations and track their movements in real time. The third key technique is patch clamping. A glass micropipette forms a tight seal with a small patch of cell membrane. This allows scientists to record electrical currents as ions move through individual channel proteins. This technique provides detailed functional data about how transport channels operate. Scientists also use advanced imaging techniques. Methods like FRET can measure interactions between proteins. Total internal reflection fluorescence, or TIRF, visualizes molecules near the membrane surface. Cryo-electron microscopy reveals detailed three-dimensional structures of transport proteins. These experimental methods have profoundly advanced our understanding of facilitated diffusion. They've revealed detailed structures of transport proteins, identified key binding sites, and established connections between protein structure and function. This knowledge has also been crucial for developing treatments for diseases related to transport disorders. Plant cells employ facilitated diffusion for essential molecular transport across their membranes. Plant cells utilize specialized channel proteins called aquaporins that facilitate water movement across their membranes. Aquaporins increase membrane permeability to water, allowing efficient hydration and maintaining turgor pressure, which is crucial for plant structural support. Plant cells also employ carrier proteins for nutrient uptake, such as glucose, amino acids, and essential minerals. These carrier proteins facilitate the selective transport of nutrients down their concentration gradients without requiring energy. Let's compare facilitated diffusion in plant versus animal cells. Plant cells have unique adaptations for facilitated diffusion. They require transport across cell walls, use vacuoles for storage, and employ plasmodesmata for cell-to-cell transport. To summarize, plant cells rely on facilitated diffusion for water and nutrient transport through specialized proteins. Despite structural differences from animal cells, they share similar transport protein mechanisms while addressing plant-specific requirements. Cellular respiration is a vital process that converts glucose into energy. But for this to occur, molecules must first enter the cell and move between different cellular compartments. Facilitated diffusion plays a crucial role right from the start of cellular respiration. Glucose, the primary energy source, must first enter the cell. Glucose molecules cannot easily pass through the cell membrane's phospholipid bilayer. Instead, they rely on specific GLUT transporters embedded in the membrane. Once glucose enters the cell, it undergoes glycolysis in the cytoplasm to form pyruvate. For cellular respiration to continue, pyruvate must enter the mitochondria. Pyruvate molecules use specific mitochondrial pyruvate carriers, or MPCs, to cross the outer mitochondrial membrane through facilitated diffusion. Beyond glucose and pyruvate, cellular respiration involves the transport of many other molecules through facilitated diffusion. ADP and ATP move in and out of mitochondria via the adenine nucleotide translocator. Fatty acids, another energy source, enter mitochondria through the carnitine shuttle system, and electrons move through protein complexes in the electron transport chain. These transport systems must be coordinated to maintain efficient cellular respiration. The rate of glucose entry must match metabolic demands. Glucose enters the cell, pyruvate moves into mitochondria, and ATP is transported out to power cellular activities. This coordination ensures that energy production meets cellular needs. Transport rates adjust to metabolic demands, and regulatory mechanisms control protein activity. Defects in any of these transport systems can disrupt energy production and lead to metabolic disorders. Computational modeling has revolutionized our understanding of facilitated diffusion by allowing scientists to visualize and analyze transport processes at the atomic level. Scientists use several computational approaches to model facilitated diffusion. Molecular dynamics simulates atomic movements. Monte Carlo methods model probabilistic events, and machine learning predicts protein function from structural data. These computational models reveal critical insights into the relationship between a transport protein's structure and its function. They help identify binding sites, channel mechanisms, and conformational changes that facilitate diffusion. For example, a model can show how a molecule binds to a specific site, passes through the channel, and undergoes conformational changes during transport. Let's examine a computational simulation of facilitated diffusion across a membrane. The model tracks molecules moving from an area of high concentration to low concentration through a specific channel protein. The simulation calculates how molecules interact with the channel protein based on physical properties like molecular size, charge, and the specific shape of the channel. These computational models have wide-ranging applications in research and development, from drug design to disease modeling and biotechnology. Despite advances, challenges remain in improving computational accuracy, integrating models across multiple scales, and validating predictions with experimental data. As computational power increases, these models will continue to provide deeper insights into the mechanisms of facilitated diffusion. Research on facilitated diffusion continues to advance in several exciting directions. One major research frontier is the determination of transport protein structures. Advanced techniques like cryo-electron microscopy are revealing how these proteins change shape during transport. Personalized medicine is another exciting frontier. Researchers are studying how genetic variations in transport proteins affect drug response, enabling tailored treatment approaches. The development of artificial transport systems is a growing field. Scientists are creating synthetic membranes with engineered channels that mimic biological transport proteins. These research directions will have significant impacts on medicine and biotechnology, from improved drug delivery systems to novel diagnostic technologies and treatment approaches. As these interconnected research areas advance, we're gaining deeper insights into facilitated diffusion and developing innovative applications that could transform medicine and biotechnology. Facilitated diffusion is a critical process that allows specific molecules to cross cell membranes without using cellular energy. This passive transport process enables molecules to move down their concentration gradient through specialized transport proteins. Facilitated diffusion relies on two main types of transport proteins: channel proteins and carrier proteins. Channel proteins form water-filled pores that allow specific ions or molecules to pass through and can be always open or gated. Carrier proteins bind specific molecules and undergo conformational changes to transport them across the membrane. Facilitated diffusion has several key characteristics that distinguish it from other transport mechanisms. It's a passive process requiring no energy, follows concentration gradients, demonstrates high specificity for molecules, exhibits saturation kinetics, and is sensitive to environmental factors like temperature and pH. The biological significance of facilitated diffusion cannot be overstated. It's essential for nutrient uptake, ion homeostasis, cell signaling, water balance, and even has implications for drug delivery and therapeutic design. In conclusion, facilitated diffusion is fundamental to life, enabling cells to maintain their internal environment while exchanging materials with their surroundings. Facilitated diffusion serves as the essential bridge between cells, and their environment, allowing life as we know it to exist.